All right. So hello, my name is Denis Torre. I'm part of the Mayan lab at Mount Sinai. And today I'll give a brief update from the work we've been doing as part of the links visualization supplement in collaboration with BioCaddy. So the overarching goal for today's work, for the work I'll present today, is to enhance DataMed by introducing new user interfaces, which will enable users to associate the datasets indexed within the repository to the computational tools which can be used to analyze the datasets and the results of the analyses themselves. So just before I begin, I'll just give a brief overview. So most of the work I'll be presenting today has been developed as a form of a Chrome extension. So I'll just give a brief couple of slides to explain what a Chrome extension is and why we decided to use this type of software. I'll then explain in detail what these interfaces accomplish for DataMed and give a brief demonstration of how they work. And I'll then round up a presentation with a couple of slides explaining the future directions of the project. So very briefly, Chrome extensions are small programs that can be used to enhance user experience in the Google Chrome web browser. They can be installed by visiting the Chrome Web Store. I have the link here. They're mostly free. They generally weigh several hundred kilobytes, and they're really easy to install. Many of them are widely used. One of the most popular ones is AdBlock, which helps users block advertisements on websites. So the idea is that these Chrome extensions can also be integrated within existing websites. So most of them are developed using JavaScript, CSS, and HTML, and they can be developed independently of the websites they end up enhancing. And they can also be easily integrated by incorporating the source code of the Chrome extension. So the reason we decided to develop these interfaces using this type of software is to allow us to work independently from the DataMed working group, and we can then converge and incorporate the results once we are happy with the final product. So before I begin, just to mention, this work is part of a larger project known as Datasets to Tools, which aims to promote the association between computational tools and biomedical data sets, and can be accessed at the URL available here. So briefly, before beginning, I'll just give a little bit of background. So one of the key elements which we introduced upon defining this project is a new type of digital object we named the canned analysis. So the canned analysis essentially is a pre-run analysis of a biomedical data set by a computational tool, whose results can be browsed online by a user and can be accessed and viewed and shared. Canned analysis is defined by 4 key features. The first of which is a hard-coded link to a web page, which contains the results of the analysis run by the tool on the dataset and can be accessed and browsed by users who navigate to the link. We then have one or more datasets, which are referenced to by means of a unique accession. We then have one or more computational tools which have been used to generate the analysis, whose results are available in this web page and are also referenced to either by a name or the tool homepage URL. And then we have one or more types of annotations, such as a title, a text description, keywords, and metadata tags, which allow users to better understand the aim of the analysis and also better index the object so that it can be more easily found. So as a brief example of what a canned analysis could actually be, so a user could download a transcriptomics assay, for example, Link's dataset LDG1297, which is an RNA-seq analysis of induced pluripotent stem cells derived from ALS and SMA patients, as well as control patients. And so the user could decide to perform a differential gene expression analysis to identify what genes are overexpressed or underexpressed in the disease cell lines compared to the controls. The user can then perform an enrichment analysis using a tool such as Enricher, which generates web pages containing the results of the upregulated and downregulated enrichment analysis. So then the results can be stored as canned analyses by adding a title, a description, and any number of customizable metadata tags. And the canned analyses can then be displayed, for example, as cards. These can be indexed within a repository and also displayed and be available to users by means of a search engine. So the idea behind this project is, behind the updates that we'll talk about today, is that users across the scientific community could generate a large number of different canned analyses by performing analyses of biomedical data sets from all these different repositories by using a variety of computational tools. Provided the results of these analyses are available online, they can be stored as canned analyses within our database, and we will then make them available directly on DataMed. So users, specifically, this project is more geared towards biologists and users with a relatively lesser computational experience. Users will be able to identify and find analyses directly from within the DataMed search pages and landing pages. So now I'll get into the actual interfaces. So the two main pages we aim to enhance are dataset search pages on the left and the dataset landing pages on the right. And so the functionality we aim to add is shown by the toolbars below the search results in the search result pages and these tables in the dataset landing pages. I'll briefly explain what is going on for the search pages, and the functionalities on the landing pages are very similar. So the idea is that we will add interfaces, and below every search result, below every data set, we'll have a toolbar. So the icons within this toolbar represent different computational tools which have been used to generate analyses based on the data set and which are stored in our database. And so by hovering over one of these tool icons, the user can visualize information about the tool, such as the tool title, the number of canned analyses associated to the tool dataset pair, and a brief description of the computational tools function. Upon clicking on one of these icons, the user can then trigger a small table which lists all the analyses which have been indexed and are accessible, as well as a brief description of the tool again. By clicking on the link available on the first column of the table, the user is then redirected to the actual results of the analysis. So in this case, it's an enrichment analysis and an interaction network of the top, I believe, 50 most overexpressed genes in a stomach cancer data set compared to the controls. And so this provides really easy and quick access to users from the data to the actual results and to actual meaningful biological analyses. Second, the user can also visualize the title of the analysis and a brief description by hovering over the middle column. There's also the options to visualize the metadata associated to the analysis. So for example, we can see the cell type, the disease type, the number of genes which have been involved in the enrichment analysis, the IDs of the samples which have been used for the controls and for the perturbation samples to perform the analysis. This metadata can also be downloaded by clicking on the download button here in either JSON or text format. And the interfaces also provide a way for users to copy the URL of the results page, which can be easily shared, and also provides a brief snippet of HTML code which can be used to embed this link in websites to promote shareability of this object. So as I mentioned before, these interfaces are available as a Chrome extension, which can be downloaded at the following link. This is, of course, free. It's several hundred kilobytes in weight, so it's pretty lightweight and simple. You're welcome to try it out. And we're currently working with a development team of the DataMed website to make these native to DataMed itself. So in this case, this is currently limited to users of Google Chrome. But in the future, once this will become native to the website, users with any browser will be able to access it without having to download any additional software. And again, as I mentioned before, this is part of a larger project known as Datasets of Tools, where we're currently indexing a large number of pre-run biomedical analyses as computational tools on datasets. So we currently have over 30,000 analyses, which have been performed on over 6,000 datasets, and we also have information of over 45 tools. And this is currently being expanded. This website is publicly available at the link here, and you're also welcome to visit it and explore our index analyses. So before concluding the presentation, I'll just give a brief overview of one of the next steps we aim to explore in the coming months, which is to add an option to users to generate on-the-fly Jupyter notebooks. So for those of you who are not familiar with Jupyter Notebooks, they are open source document. This is an open source platform which allows you to create interactive notebooks containing live code in over 40 languages. It also supports Python, R, and many other very popular languages in data science. It also supports annotations to explain what the code does and what the aims of the analysis are. And it also supports multiple interactive plots, which can provide interactive ways for users to explore the data and the analyses. So for example, this screenshot here shows an interactive heat map, which has been developed using cluster grammar, which can be interacted with to explore the data itself. These can also be viewed as an HTML web page, which is hard-coded URL, so you can also easily share it among the scientific community. And they can also be downloaded and run locally. So users are actually able to rerun the code which has been used to generate the analysis and to tweak it according to their needs. So the general idea behind this new direction is that we want to develop an API which will consist of several scripts which go into the repositories of these different data sources and download the data sets and make them available in a standardized format in the Jupyter environment. And on the other hand, we will also aim to develop or collect several scripts which allow these computational tools to operate on these standardized data formats and generate meaningful analyses. So this API will also support a configuration as a form of a JSON file. And this will allow users to create interactive and tailored Jupyter reports of the data sets they're interested in analyzing by a combination of the computational tools of choice. From an interface perspective, we aim to implement this by adding a small button on the right-hand side of the toolbars. So this plus just represents the user can create a new analysis. And this is still a mock-up, but we're working on developing this interface. So the user will be able to access this sort of tool marketplace, where he will be able to select the tools of choice to generate the analysis. You'll be able to add or remove data sets, to set different settings in order to parameterize and set up the analysis in a way that he wishes. And then by clicking on the Continue button, this will generate one of these Jupyter notebooks, which will be either available to download and to run locally, or which will also be available online to access, to view, and to share. So this wraps it up. I'd like to thank you for the attention, and I'm happy to take questions.